project. And what you're looking at on the screen here are just some pictures of different organic amendments like biochar. This is compost. This is, I believe, rice bran that we apply during the process called anaerobic soil disinfestation, some wood chips or mulch, another picture of anaerobic soil disinfestation, and some vermicompost. And I'll try to hit upon all of these different types of organic amendments during the talk. So this is just an outline. I just like to give a brief introduction to my research program, and then talk broadly about organic amendments, their mechanisms of disease suppression, effects on soil properties, and the benefits. And there are potentially some trade-offs of using organic amendments. And then I'd just like to present some of the research that we've done here at Davis in my lab and with collaborators, looking to identify effective carbon substrates for ASD, and some more recent work that I have in collaboration with Anna Pastrana-Leong, looking at the survival of Fusarium, oxysporum, which causes Fusarium wilt in ASD-treated soils. And so Hung introduced me really well. So I'm a soil microbial ecologist, and most of my work basically tries to understand how soil microbes contribute to plant and soil health. And so pretty much all the questions that we ask in my lab have to do with like what management practices, how do they affect soil microbial communities, and specifically how do these practices affect who and what is present in the soils. And to me, one of the most important questions is understanding the how and why microbes are doing particular things. And so what we're interested in my lab is understanding the role of microbes in controlling soil-borne plant pathogens, and also building soil organic matter. So the two main projects that I have focus on anaerobic soil disinfestation and whole orchard recycling. But I also have some collaborations with deficit irrigation work in tomatoes and looking how it interacts with disease, and have a list of just some projects that we have. I've been working with a startup company called 2 Biomics, which has developed this microbial extract that exhibits fairly good suppression against plant pathogens. So that's been some interesting work. And I also worked on some genomics of bacterial plant pathogens, and we are starting a project to look at biocontrol of Agrobacterium tumefaciens. And as you can see in this picture, I actually mainly work in tree crops, not vegetable crops, with the exception of my deficit irrigation work. And so, we've been trained to adapt ASD for tree crops, and specifically for the treatment of replant disease in almonds. And so a lot of my work is focused on that. And then for whole orchard recycling, what I've been mainly interested in is trying to figure out mechanisms of organic matter accumulation in the soil. And I do have an interest in trying to see, because a question that frequently comes up with that process is, does it affect the survival of wood decaying pathogens in soil? And that's not an answer that we don't have a clear answer to that question yet. So this is what's going on in my lab. So if you're interested in anything, feel free to contact me. So going into organic amendments. When we think about organic amendments, we're typically thinking about compost. So it can be compost, vermicompost, which is made with worms, composted manures, biochar, sometimes people just mulch in, wood chips or crop residues. And I also include biostimulants, sort of inoculates, whether or not they're single strains or actual communities under organic amendments. And typically when we're adding an organic amendment to soil, and this is admittedly a microbe-centric view, is we're trying to alter the soil microbial community. So we want to affect some change in the composition and the function of the community so that we can get an outcome. And in this case, we want to have disease suppressive soils. And in general, there are 2 types of disease suppressive soils, and they are actually often found in the same soil, but there's a concept of having general suppression, which is where soils basically inhibit the growth of plant pathogens. And this is due to the collective, competitive, and antagonistic activity of the entire soil microbiome. And we can also have specific suppression, which is actually due to the activity of specific strains or groups of microorganisms. And in general, it's thought that if you have specific suppression, which is due to a particular species or group, that you can transfer this suppression activity between soils by simply inoculating soils with the strains or consortia of bacteria or other microbes. But general suppression is thought not to be something that's transferable between soils. And so when we think about sort of what organic amendments do, and there's a lot of studies on organic amendment effects on microbiomes and other biological characteristics in soils, and some of the frequent observations that we see is that it leads to an increase in microbial biomass. You're adding a carbon source to soil, so microbes are able to consume that carbon and build biomass. It tends to increase microbial diversity as far as who is present, so the taxonomic composition of the community. And there's increasing studies showing that it actually affects the functional diversity of the soils too. And so this is just a figure from a paper of from Dan Klipfel's lab and a postdoc who we had several years back now, who looked at the effect of a vermicompost on soil microbial communities and compared that to the unamended soils. And then these were soils that were fumigated with 2 different fumigation techniques, and then soils that were vermicomposted. And what I'd like you to take away from this figure is that, you know, the more colors we see and the different sort of distributions of bars, that is indicating that we have more diverse communities under vermicomposting. And what's interesting about it is these last 3 bars here are actually soils that have been mixed with vermicompost after fumigation. So we've basically reestablished community diversity by mixing fumigated soils with vermicompost. And I don't know if I have it in here, but one of the things that they were looking at in the study was suppression of agrobacterium by vermicompost, and it's super effective at doing so, at least in these soils. And part of the reason why we think that increasing microbial diversity is that organic amendments increase microbial diversity and suppress plant pathogens is basically we're increasing the number of organisms in the soil that can compete with pathogens. And so you kind of get this, this is from a paper describing a positive feedback between increasing diversity, increasing competition, increasing antagonistic populations, and leading to pathogen suppression. And a lot of times people have tried to either through predicting functions or actually testing, isolating and testing microorganisms, they have identified a number of microbes with biocontrol or beneficial or plant growth promoting functions that frequently go up in abundances in organically amended soils. And this includes trichoderma, fluorescent pseudomonads, actinomycetes, specifically a lot of streptomycetes, a lot of bacillus species, some non-pathogenic Fusarium, which can exhibit biocontrol. So in general, you're basically providing the soil community with various carbon sources that opens up more niches, so you can have greater diversity of microbes, and they can, you know, with that greater diversity, you're introducing potentially more functional diversity and competition for plant pathogens. So when we think about mechanistically why organic amendments work, it's actually, they're essentially doing biofumigation of the soil. So instead of injecting a chemical fumigant, which are all volatile organic compounds, you're essentially making those volatile organic compounds in situ. And they typically suppress or kill the plant pathogens, so they can be static, they're preventing their growth or activity, and sometimes they actually affect their cell wall structures and things like that. So, and one thing about a lot of the volatile organic compounds that are produced, a lot of studies have demonstrated that they're really effective at controlling plant parasitic nematodes, which I know can be difficult to control. And there are a ton of studies in VOCs, and there's a very long list of the ones that are produced, but they kind of fall into these categories of having sulfur involved, being benzene derivatives, and different forms of alcohol, organic acids, and things like that. And what's interesting about this is that, VOCs can actually be produced by a single strain that's producing a VOC that's inhibitory or killing to plant pathogens, or it could involve a consortia or a group of microorganisms producing compounds that are inhibitory. And this actually opens it up to an interesting question that a lot of people in sort of the microbial ecology world are pursuing, is basically constructing communities that can produce compounds specifically to kill pathogens or do some other function, and they're called synthetic communities. So that's a line of research that's going on. And besides this production of volatiles, we also see other functional changes in the soil. So there's been some studies that have demonstrated antibiotic production. One thing that's frequently observed is an increase in chitinolytic enzyme activities, which is actually pretty good at, because plant parasitic nematodes have chitin in their eggs shells, and this activity actually can lead to killing of those eggs. And so to me, the action of these organic amendments is the stimulation of microbes to basically carry out a biofumigation of the soils.
Misha, we have a question from Mike. If you don't mind.
Yeah.
Mike asks, how does red seaweed inhibit methane production?
Oh, I do not know how red seaweed inhibits methane production. I could look that up. My intuition is that, so if you're doing methanogenesis is an anaerobic process, and so I don't know if it's affecting the redox potential of the soil in some way that's preventing it. It could be a direct actual inhibition of methanogens, because there's a very specific group of organisms that are capable of producing methane. But I can look that up and get back to you, Mike.
Thank you, Misha.
I noticed there's a question about trichodoma and actinomycetes and optimal soil temperatures and soil types. That is something else that I would have to look up. But I will say that microbes generally have, even though if they have an optimum, they usually have a broad range of conditions over which they can survive. So, you don't have to be exactly at that sweet spot in order to have them be active. And so another way that organic amendments can basically help suppress plant pathogens is sort of indirectly. So when you're adding organic amendments, you're also affecting the soil physical chemistry. And one of the most important things that you're doing is building soil organic matter, and that has implications for other properties in the soil. So it tends to be associated with increasing soil fertility, retention, and I have on another slide other important outcomes of increasing soil organic matter and improving soil health with organic amendments. And when you look in the literature to see sort of what are the sort of findings of the effect of organic amendments on specifically pathogen suppression and how that interacts with the soil physical chemistry, there's actually a little bit of a varied response. But I did find a study that conducted a meta-analysis that found that soil properties could explain about 60% of the variance in crop yields and organically amended soils. And this is what this is showing up here. So basically, even though the microbes are certainly doing a lot, we still can't overlook the impact of organic amendments in soil physical chemistry. And of course, if you're improving soil health, you're likely improving plant health. So a lot of times, if you have better soil fertility, you can basically increase your plant nutrient status. There's been a few studies showing that plant immune responses are actually responsive to organic amendments. And so that can also sort of indirectly make the plants more healthy, so they're more resistant to pathogens. And finally, there's been some work showing that when we apply organic amendments to the soil, that you can get ammonia volatilization that can actually suppress plant pathogens too. And this may not be, it could be due to biological activity, but it might also be a pH effect too. because as pH increases, you lose more ammonium. So apologies, my nose is running. So getting to the benefits and trade-offs of organic amendments. So I think I've listed a lot of the benefits, but essentially we're expecting that with organic amendments that over time, ideally you should be able to reduce reliance on mineral fertilizers if you're increasing nutrient retention in the soils. Generally, you should expect crop yield increases. Again, we often see changes in the soil physical chemistry, so increased pH, soil structure improves, aggregate stability is related to soil organic matter, connects soil pores, increases infiltration rate. You actually can get greater penetration of roots into the soil, so there's all sorts of improvements to the soil properties that happen with organic amendments. And I think we're fairly confident that they have a positive impact on microbial communities in the soil and the rhizosphere, which are microbes kind of most directly associated with plants, and that they can impact plant disease. And another thing is it's a very good way to like manage farm waste. And a lot of, I know the Almond Board has a zero waste initiative. So getting to the point where we're using as much of the material that we're producing or we're purchasing on the farm is, I think, a good goal. But there are some trade-offs or considerations that you can think about when you're deciding on an organic amendment. So it's not super common. And actually, I think it happens mostly with studies in biochar, because I think it really does take a long time to see effects of that amendment. But there are some out there that show that you really don't have an effect of organic amendments in disease suppression or plant growth. So, you know, it's kind of like in these situations, it may not be harmful, but it may not have the benefits that are expected. Another thing is like, so you can apply too much organic amendments, especially compost. So one thing that can happen is you're dumping a bunch of carbon into the soil. That's gonna, once you imbalance the carbon and nitrogen ratio, you're probably gonna lead to a lot of CO2 being respired, and that's a greenhouse gas. Another thing that can happen is if you have soils that are especially high in organic matter, which sometimes putting more organic matter into the soil actually primes decomposition of the native soil organic matter. Seems counterintuitive, but that can actually occur where adding carbon to a soil that's, I don't want to say saturated with carbon, but you're essentially, because the compost or other materials that you're adding are assumed for the most part to be a little bit more labile than the native soil, you can actually prime that release of soil organic matter that has accumulated. Another thing with excessive application is, you're overloading on nutrients, which can leach into groundwater and cause all sorts of issues. Another thing is a lot of the organic amendments are derived from like animal waste, and if they're not properly treated, you can actually spread pathogens. And there is some work on looking at the spread of antibiotic resistance genes through manure, so that's also a concern. And I think importantly, the cost of it. So, and do you need any sort of specialized equipment? It would probably be good to find locally available organic amendments. So those are all sorts of considerations. But I think overall, I think that they likely benefit and probably don't hurt. So now I'm just going to get into some of the research that we do in my lab. And so anaerobic soil disinfestation is basically a pre-plant organic amendment. And it's been developed as an alternative to the use of chemical fumigants, which are basically given regulatory constraints and just urbanization, the future of using these chemicals is uncertain. Like few of them are being registered. Methyl bromide is completely phased out, which was super effective at treating pathogens. And so people have looked into ASD, which was developed in the late 1990s, early 2000s in Japan and the Netherlands at around the same time, to actually replace chemical fumigants. And during the process, you basically spread a carbon source that's labile, and ideally something that's locally available. And so onto the soil surface. And one of the gold standard carbon sources is rice bran, and it's typically applied at 20 tons per hectare. And once you have your carbon source spread out, you basically till it into the soil. And the soil then can get covered with a gas impermeable tarp. And also under this tarp, there are irrigation lines, because once everything's set and go, the soil is going to be flooded or irrigated to filled capacity. And then you can leave the soil like this. And typically we're talking about 4 to 6 weeks. I've seen as long as 15 weeks leaving the soil covered. And what that causes is basically a change in soil conditions that lead to pathogen suppression. And ASD has been used in a variety of crops. I know it's used in strawberry production here in the Southeast. A lot of vegetable growers use it, and it has been shown to be effective against a number of plant pathogens. And it's considered broad spectrum, so it can be effective against bacteria and fungi and mycetes. It has been shown to be effective against nematodes and weeds in some studies. The problem with nematodes is they can migrate. So when you're treating the soil with ASD, you're essentially targeting mainly the sort of upper foot of soil, and nematodes can migrate below that. And the weed suppression is very clear after implementing ASD. You can just tell with soils that have been treated, but it dissipates over time. So it's not like it's going to last past that initial treatment. And this is just a figure from a really nice meta-analysis paper that's available in Frontiers, that's free to download, that basically looked at the literature and basically put all of these data together to look at the whether or not ASD is effective at suppressing a plant pathogen. So on the scale, if it's a negative number, it's actually ASD is suppressive. And for a lot of groups, ASD is pretty effective at controlling plant pathogens. And it, fungi, there's a little bit of a spread for some of them, but it's so it's broad spectrum. It tends to work. There is a need to optimize it and test it for various conditions. Like it may work better in the summer than it does in the winter. It's carbon source dependent, so there are some things like it's not one size fits all, but it generally does work. And so the way ASD works is it basically alters the physical, chemical, and conditions in the soil, and also the soil microbiome. So one of the things that happens with ASD is that you're covering the soil with a tarp, and so you're actually getting a little bit of a heat effect. So you can actually combine ASD with solarization too, where you can increase the temperatures of soil to get more effective pathogen control. Of course, soils go anaerobic. So this is a figure showing redox potential in soils and over time. And you can see that what happens is with the carbon source added to the soil, the microbes consume that carbon, and when they're consuming that carbon, they consume the oxygen, so that lowers the redox potential. We also tend to see soil pH decrease, and one mechanism that was identified probably very early on in ASD studies is that these physical chemical changes in the soil release metal ions that also seem to be inhibitory to plant pathogens. But one of the biggest things that occurs in the soil is we get this shift in the community towards anaerobic microbes, specifically those belonging to the phylum Firmicutes. And basically, these are the organisms that mediate pathogen suppression by producing volatile organic acids. And there's been a lot of short chain fatty acids which have been implicated. So these are things like butyrate, acetate, lactate, which can have been shown to be effective at inhibiting plant pathogens. So they're basically fermentation products that accumulate in soils and have some ability to suppress or kill plant pathogens. So this is just a figure from Sarah Strauss's work in Dan's lab, showing that ASD can significantly reduce populations of Agrobacterium tumefaciens. So this is their abundances before ASD at different soil depths. And we can basically see for all of these different soil depths, we are able to recover agrobacterium until we get to about 18 to 30 inches. And this is just because ASD works best in the upper sort of 12 inches of soil, just because of the way it's performed. You may be able to increase its efficacy at soil depth by burying irrigation lines at lower depths and using a liquid carbon substrate that can have it penetrate further into the soil. And so, based on a lot of work in Dan's lab and also Greg Brown's lab, we have some research questions on ASD. And one of them is, so we're working in tree crops, and ASD has really not been used in like almonds and walnuts. And part of that is the cost of doing it. So you can imagine there's a tarp involved, which is pretty expensive. The carbon substrates vary in expense. And so we've been interested in understanding, can we reduce the cost by identifying cheaper carbon substrates, and specifically agricultural byproducts that are locally available. I have an interest in understanding the effects of the different carbon substrates in soil microbial communities and identifying microbial taxa that consistently occur across ASD treatments, because that could be sort of a core ASD microbiome that we're eliciting potentially in soils. And I'll show you some data for these first two questions. Actually, I don't think I included anything on this last question. And so looking at the cost of the various substrates, so this is a figure from Greg where he compiled, mustard seed meal is a very effective ASD substrate because it produces, so when it decomposes, there's isothiocyanates that get produced, which are really effective at controlling plant pathogens. But it tends to be very expensive, and even though it's a great substrate, the cost of it really does kind of make it out of the reach for most people to actually buy and use. Rice bran is the gold standard. It's ASD was, I think, originally developed either with rice bran or ethanol. And we've also been looking at like almond hulls and shells, tomato pumice, other grape pumice, pistachios. So all of these different agricultural byproducts, which decrease in cost and hopefully can make ASD just much more easier to implement. And so in order to first ask this question about like, okay, can we screen agricultural byproducts and identify cheaper substrates? So there was a postdoc who's now at the CDFA, Sebastian Albu, who conducted some greenhouse studies, and he looked at 16 carbon sources and basically inoculated agrobacterium into the soils and looked at the recovery of agro and also measured redox conditions. And my lab followed up with some microbiome profiling of some of these soils. And so we're using the recovery of agro and achieving lower redox potentials as the measurement of the effectiveness of a carbon substrate. And so this is just showing agrobacterium counts on the y-axis. On this y-axis, on the left, on the right, this is cumulative anaerobicity. So what the redox potential, in the soil. So the bigger this is, the lower the redox potential, the more anaerobic the soils were. So this is over time. And then these are the different carbon substrates that were tested, and controls are simply untreated soils. And so we can see that untreated soils, agro is very happy. It survives just fine. And we've arranged this so that we go from not very suppressive or not very good ASD carbon substrates to very good ASD carbon substrates. And so we can see that we have a group down here that pretty much either almost completely kills or eliminates agrobacterium timofasciens. So these are sort of substrates that we've identified as being potentially very good elite substrates. And so that's based on just the agro counts. And if we look at the cumulative anaerobicity, there's a nice relationship here, and that substrates that are really good at killing agro also lead to lower redox conditions. So that's likely related to the shift in the communities. They're producing those volatiles that are basically inhibitory to agrobacterium timofasciens. And so one of the things that we can ask ourselves, okay, so we have these two different groups of carbon substrates, some that are very good, others that aren't. Sort of what is it about those carbon substrates that like that sort of, is there anything chemically about them that can sort of lead us to suspecting which ones are better? And this is just a principal component analysis of the chemical composition of the solid ASD substrates that we use. And basically the message here is that when we look at this first PC axis, our soils that are less suppressive are basically grouping over here, and our soils that are more suppressive are grouping over here. And what they tend to have is a lower C to N ratio and a higher nitrogen and phosphorus content than soils that are less suppressive. So it's telling us that nitrogen and phosphorus content and a lower carbon to nitrogen ratio, identifying substrates with those characteristics might be a better way of, you can screen them, of course, in a greenhouse, but you could probably look at the composition of a potential substrate and say, okay, this looks promising because it has a low C to N ratio and high phosphorus content. So it's just something that we're looking into more about, doing further characterization of these substrates to really figure out, okay, what really groups the ones that are effective versus ones that aren't. And so for looking at microbial community changes, my lab did is we came through and we kind of looked at all of them, and we were like, okay, let's work with rice bran and a couple of the other substrates that are effective. And then I was interested in red grape pumice because it does suppress agrobacterium, but it doesn't give the same level of cumulative anaerobicity. And so I was kind of interested in saying, okay, is this potentially because it elicits different microbial communities? And just following up on a substrate that, is pretty effective, but also doesn't necessarily result in the as low of a redox potential as other So it has good redox potential, but it's not completely eliminating agro. So that's why I was interested in it. And this is just, so what we did, we sequenced bacterial communities in soils. And this is just a way of showing the community composition of our samples. And so in this diagram, samples that are close together are similar in community composition. And when we did this work, we did it in a greenhouse, and we sampled our starting soils, which is this plus symbol here. And then we sampled at 2, 9, and 21 days into ASD treatment. So those are the circles, the triangles, and the squares. And we had a no carbon control rice bran, tomato pumice, and red grape pumice in this particular study. And so we see over time that we see this clear shift in the communities for the ASD treatments, but not for our controls, which is nice because we aren't seeing, these soils were anaerobic, but the communities basically stayed very similar over time. And what's interesting about this is that we have tended to find that rice bran and tomato pumice are pretty similar to each other with efficacy against agro. And what's nice about this is that their community compositions also track. So rice bran is the blue and tomato pumice is the orangish color. And so in space, these communities are grouping together, which means that they have similar compositions. And then when we have our red grape pumice, which is the green, that was the substrate that did go anaerobic, but was not as suppressive as rice bran and tomato pumice against agro. And we do see that there's a difference in the community structure between red grape pumice and the other two substrates. So that, to me, points to, okay, there's something about the community that red grape pumice stimulates to grow that isn't as suppressive to agro. And it's something that we can follow up and trying to figure out, okay, what is different about that community, or what's special about the rice bran and the tomato pumice communities that lead to pathogen suppression. And so just to summarize that work, so I personally liked tomato pumice as a carbon substrate for ASD. It's not too expensive. I like the way it smells in the field. And it gives us pretty similar effects as rice bran. Some additional directions that we're pursuing. So Greg Brown has a very much interested in figuring out if we can also lessen the cost of doing ASD by removing certain components. So, we definitely need the carbon source, I think, but do we necessarily, maybe some of the effects of ASD that we're seeing are nutritional rather than just pathogen suppression, and this might also depend on soil type. That plastic tarp is expensive and it gets disposed of, so it's kind of wasteful. So, how necessary is the tarp? And so these are things that we're pursuing in field trials. One of the things I'm interested in is figuring out, okay, how does ASD affect soil nutrient dynamics beyond the incubation period? And there's been some studies in the Southeast showing that depending on the carbon substrate, that you can increase nitrogen content in the soils. And one thing that I do see in our data sets is that we see an increase in nitrogen fixing organisms during ASD, and it just could be the fact that into fixation actually occurs under anaerobic conditions, so the organisms that tend to survive have those into fixing genes, but it's something that we're also going to pursue. And we're interested in how ASD works with other management practices. So we've done some studies integrating ASD with whole earth recycling, what happens after ASD, and applying compost or cover crops or doing things like that. And I'll use my last few minutes here to present a little bit of data that we have on survival of Fusarium wilt pathogen in ASD-treated soil. So this is in collaboration with Anna Pastrana-Leon. She's a plant pathologist who actually knows all about Fusarium oxysporum, and this causes Fusarium wilt of strawberry. And it's actually a very difficult pathogen to treat, is my understanding. So it can persist in symptomless rotation hosts. It's a competitive saprophyte in soils and can colonize crop residues, and it can survive ASD and other disinfestation methods. So the fact that it can survive ASD is related to the fact that it can survive anaerobic conditions, and there might be some other things contributing to Fusarium survival in the soils. And so there's been some studies on the effectiveness of ASD against Fusarium, and none of them have come to a consensus yet, but so it tends to depend on temperature. The carbon substrate does seem to matter a lot. How long you stay anaerobic can affect the how effective ASD is at reducing Fusarium populations. And so because of this, Anna had an interest in actually understanding, okay, so let's say that we have an ASD-treated soil and we inoculate Fusarium. So the Fusarium hasn't actually undergone ASD. Okay, what happens to the Fusarium that's inoculated? Like, does it survive better or worse than an untreated soil? So she set up a greenhouse experiment where she had untreated control soils and then ASD performed with rice hulls and mustard seed meal. And part of the reason why she added mustard seed meal is because there's been some previous work saying that mustard seed meal is probably one of the better carbon substrates for controlling Fusarium oxysporum. And so she set this up so that she had 6 replicates under each treatment. She performed ASD, collected the soils post-treatment, and then set up another 6 replicated sets of mesocosms where she had the soils and inoculated Fusarium, and then collected soils at 1, 3, and 7 weeks post-inoculation. And so she did this experiment 3 times. And so, so basically we have a replicated experiment, which is nice to see how consistent the response is. And so basically she counted Fusarium oxysporum populations. My lab did the microbiome profiling of fungal and bacterial communities, and also measured some soil physical chemical properties. And I'll just talk about the first 2 points here. I'm still actually looking at the soil physical chemistry data. And so, so what she found across her experiments is, if you're repeating experiments, look for consistency. And what she found was, like, in the 2nd and 3rd experiment that she conducted, the Fusarium populations were lower in ASD-treated soils than in the controls. But in experiment 1, they had similar population densities. And we think this is because when the ASD performed in this particular experiment was only anaerobic for 10 days of the 21 days, whereas here in both of these experiments, the soils stayed anaerobic for about almost the full 21 days of the ASD treatments. So we think that's what's accounting for these differences. And we can look, so this was a way of enumerating Fusarium populations via culturing. And we also just looked to see sort of what do we see in our sequencing libraries about Fusarium abundances. So we sequenced our samples and we identified this ASV, which stands for amplicon sequence variant number 12, that is likely it's taxonomically assigned to Fusarium oxysporum, so we believe that's the organism that was inoculated. And this is just a figure showing the relative abundance. And so circles correspond to basically 1 to roughly 25 up to 75% of the community. And each one of the treatments for each experiment. And so what we're finding here, and then the weeks after inoculation are on the x-axis. So if we look at each one of these experiments, we can see that the circles for the untreated control are generally bigger than the ASD treatments, and they're basically present, except for experiment 2, from our post-ASD soils. And we can see the circles in our ASD treatment are smaller, so the abundances of Fusarium oxysporum based on sequencing are lower in ASD-treated soils. Unfortunately, when you do a lot of sequencing, you get a lot of taxa where you actually don't know their identity very well, which is why we have all of these uncultured fungi here. But there is this ASV, schizophysium, I'm probably saying that completely wrong, that tends to be abundant in the ASD-treated soils and not present in our controls. And this is something interesting to follow up because there are some papers that have indicated that this is a potential beneficial fungi. And so it will be interesting to sort of look more at, tests to see if there are significant differences between treatments, and these are just plotting the 20 most abundant to see if there may be other strains of this particular species in the soil that also show this pattern. And this is a work in progress, so we have soil nutrients data that I'm slogging through and trying to relate to the Fusarium abundances. We also have data on the bacterial taxa that I need to look at, and we want to compare this because there's been a few other studies in survival of Fusarium in soils, and just sort of compare results to other studies, especially if they've done any sort of microbiome analysis of them. And I think I'd just like to thank everyone for listening to me and acknowledge my lab members. So Holly Forbes is my lab manager, and had lots of undergrads and previous lab members who have contributed to this work. I also work with Ina Gonzalez-Orta at California State and Sarah Strauss, who did a lot of work in Dan's lab on agro and vermicompost and ASD before I even arrived. So thank you.